Recording in
commercially supplied
progress.
services mission to the International Space Station contracted by NASA. I'm Patrick O'Neill, Public Affairs and Outreach Lead for the International Space Station National Laboratory, and I'll be your moderator for today's event. Okay, so let's talk some logistics for this launch. First and foremost, as mentioned, this will be NASA's SpaceX CRS-30 mission to the orbiting laboratory. The launch is slated to take place from Space Launch Complex 40 at Cape Canaveral Space Force Station in Florida in mid-March. SpaceX's Dragon spacecraft will be filled with more than 6,000 pounds of critical research and supplies and will launch on a SpaceX Falcon 9 rocket as part of its approximately 30-day mission to the orbiting laboratory. As you might have heard or read, sorry, I can't read right now, I just said the magic word of research, which is what we are here to talk about today. So today we are focusing on just a few of the investigations launching that will be supported by the on-orbit crew during this mission. We have an exciting lineup of speakers showcasing the diversity of experimentation that the space station enables. These representatives will provide an overview of their project and answer questions from media who have registered in advance. For the general public following this live and wishing to ask questions of research teams speaking today, I encourage you to do so via the hashtag ISS National Lab. But before we hear from teams launching investigations on NASA's SpaceX CRS-30 mission, we'll hear from a few representatives from the ISS National Laboratory and NASA's ISS program. For media, after these remarks, I will open to any questions. To ask a question, please use the raise your hand feature within the Zoom platform, and we will unmute your line so that you may ask your question. Or you may ask a question within the Q&A box. Again, similar to the public, to ask a question, please use the hashtag ISS National Lab. Okay, so I think that should put us in a pretty good place as far as logistics are concerned for today's webinar. So what do you say we start talking about some space-based R&D? To kickstart the conversations, I would like to invite Dr. Davide Marotta, Science Program Director in Space Biomedicine for the ISS National Laboratory, to provide some remarks. Dr. Marotta, the floor is yours.
Thank you, Patrick, and good afternoon. It's great to be here today as we discuss some of the science and technology demonstrations launching on SpaceX CRS-30. But before we get into some of the project launching on this mission, it's been already a busy year on station, which I'm sure Heidi will talk a little more about as well from a NASA perspective. In January, Axiom Space, in coordination with SpaceX, launched its third private astronaut mission, wherein the ISS National Lab was proud to sponsor more than 20 investigations during this 2 weeks science expedition. One of those projects was from the National Stem Cell Foundation, who will also join today's webinar to highlight another investigation launching on NASA's SpaceX CRS-30 mission. Last month, Northrop Grumman launched its 20th commercial resupply services mission through NASA, also including more than 20 payloads sponsored by the ISS National Lab. Now, we are gearing up for this upcoming mission from SpaceX, once again jam-packed with research sponsored by the ISS National Lab. The investigations on this mission span a variety of research disciplines. That said, there is a strong push on multiple biomedical investigation, which is near and dear to my heart as a molecular and cell biologist by training. One area that has been very promising recently has been leveraging the orbital lab to further in space manufacturing concept, including bioprinting on space station. Like Redwire Space, a commercial service provider to the ISS National Lab, will continue their pursuit on bioprinting on the space station through this mission. As many may remember, last year, the company and its research partners successfully printed tissues associated with human meniscus on the space station and then brought it back to Earth for analysis. Now the team will attempt to print a cardiac tissue in space and bring it back to Earth. Also, pharmaceutical leader Eli Lilly and company will continue their research and development on the space station through this mission. Over the years, the company has launched a series of investigations intending to create more effective therapeutics for patients on Earth. And on this mission, they will work alongside Redwire to leverage the pharmaceutical in-space laboratory. I mentioned earlier NG20, and the research team from University of Connecticut launched a project on that mission funded by the National Science Foundation, looking at cartilage repair through tissue engineering concept. Now, The team will build on that project with Axiom Space to evaluate drug delivery processes for cartilage repair that might help to reduce inflammation. That said, it isn't all life science in this mission. There will be technology development payloads, project leveraging the outside of the space station, student projects, and many more. Many of which will be highlighted today in this webinar, but I also encourage everyone to look at the ISS National Lab website, issnationallab.org, to learn more about the science flying of the SpaceX CRS-30. With that, again, very excited about this launch. A lot of innovative science will be flying, and looking forward to hearing from some of the research teams launching payloads on NASA's SpaceX CRS-30 mission.
Thank you very much, Dr. Marotta. Again, to your point, there's a lot of great science that's flying on this mission that the ISS National Lab is fortunate enough to sponsor. But to your point, also, I would encourage those that are following along to visit issnationallab.org to learn more, as we will next week be putting forth a series of features that highlight some of the investigations that are launching on this mission. Okay, next up, I would like to welcome Heidi Paris. Heidi is the Associate Program Scientist for NASA's International Space Station program and will provide a perspective from our nation's space agency.
Outstanding. Thank you, Patrick. Well, good morning, good afternoon, everybody. Thank you so much for joining us today for this webinar. We are super excited to share about some of the research that is going on board the space station, as well as some of the research that's coming up on this SpaceX 30 mission. First, I just want to give a huge thank you to the ISS National Laboratory for organizing and for hosting this webinar event. And also to all the researchers who have, taken the time out of their very busy days, very busy lives to help us understand about some of the science that they are involved with. So as Davide mentioned, it has been a very busy, a very exciting couple of months for science on board the International Space Station. First, we did welcome the third U.S. private astronaut mission to the ISS. And then that was followed closely by arrival of a new big batch of science on NG-20 that the crew has been working on for the last couple of months. And then just a few days ago, we saw the Crew 8 vehicle bring up the next group of long duration residents for the space station. So not only are they going to be conducting the research for hundreds of scientists all around the world, but they themselves are also going to be subjects for human research studies. So they're going to help us understand how spaceflight impacts the human body so that we can help to solve those human challenges that will enable future long duration space exploration, first to the moon through the Artemis program, and then beyond that to Mars in the future. So in between all of those spacecraft arrivals, spacecraft departures, the schedule has been pretty packed with support for dozens of different experiments every day. Some highlights of things that have occurred over the last couple of months. We've seen demonstration of technologies that may help enable development of remote surgical capabilities, which could be extremely important for long duration space exploration in the future, but can also be really helpful for remote medical applications on Earth. We've seen the completion of a 5-year study that is testing out the on-demand production of vitamins and other nutrients. And then just within the last couple of weeks, we have seen the successful demonstration of the ability to manufacture Z-band optical fibers in microgravity. So these investigations join over 3,700 different scientific studies and technology demonstrations that over the lifetime of the ISS, they have taken advantage of the very unique capabilities we have on board the ISS to advance, entire fields of research in a way that we can't do anywhere else on the ground. And one of the critical outputs from that research is the publication and the sharing of the knowledge that's gained with other research teams and with industry so that we can build upon those results and use them in ways to improve our lives on Earth in very tangible ways. So to date, we've seen over 4,000 different research publications come out based on that ISS research. Many of those have been in top-tier journals with high impact to the broader scientific community. And you are in luck because we, just a couple of weeks ago, published a compilation of recent results over the last year or so. And that's available now. It's called the 2023 Annual Highlight of Results. It's on our results website at www.nasa.gov/station results. And just a few highlights to kind of whet your appetite. We delve into some new insights into how the human brain adapts to extreme conditions. New discoveries about robotic mobility in space that are based on use of our free-flying Astrobee robots on board the station. And also an improved understanding of the property of foams. Foams are products that are used, they're in many of the products that we use in our everyday lives, things like detergents and food, medications. And so this type of research can help us potentially improve these products in the future. So now let's talk just a little bit about some of the science on SpaceX 30. So Davide mentioned, SpaceX 30 is packed full, right, of the hardware and the samples that will support a very diverse set of investigations, not just from NASA and the National Lab, but also across the international partnership. I'm really excited to hear more about, especially the National Lab sponsored investigations from our panel today. I'm not going to steal any of their thunder by talking about their research, but just to highlight a couple of the other investigations that we do have coming up on this flight. We have an investigation looking to understand how to create more efficient solar cells through the very precise arrangement of charged nanoparticles. We have an investigation studying the effects of microgravity on photosynthesis and on the metabolism of plants that might help support development of a bioregenerative life support system. We have a CubeSat that's sponsored by the Canadian Space Agency. It's going to use reflected signals to measure sea ice parameters that will help enhance our knowledge of ocean phenomena. And then from the European Space Agency, we have the last in a series of 6 investigations that's been performed, I think, over the last 6 years on ISS, that is using a transparent organic material to observe the mechanisms that occur as materials are solidifying to help us understand how to improve those materials processes. So if you'd like any more information on any of the experiments that you hear about today, in addition to the ISS National Lab website that's been mentioned, another great way to find out about those investigations is to visit the ISS Research and Technology webpage at www.nasa.gov/ISS-science. And you can also follow us on X at ISS_research. This is a great way to get updates on research as it's being performed. With that, Patrick, back to you.
Fantastic. Thank you so much, Heidi. At this point, if there are any questions from our friends in the media, please feel free to raise your hand or use the Q&A box. If not, then we will move on to the researchers in today's webinar. And again, as a reminder to the general public, if you would like to ask a question during today's webinar, please use the hashtag ISS National Lab. I see that we have a hand up from Jonathan Suri.
Thanks so much for taking my question. My question is probably for Heidi. I wonder if you could go a little bit more into the remote medical applications. What are the most likely scenarios that you would be studying for a long duration space flight? And what do you also see as the most likely applications back on Earth, such as in a rural or remote setting on Earth? Thank you.
Absolutely. Great question. And honestly, you sort of answered a lot of it as you were talking from my standpoint. So this investigation was basically looking to see if we could have surgeons on the Earth performing, medical-like operations on board the space station. They weren't using, actual live tissue or anything like that. They were using basically rubber bands to simulate what those organ tissues would be like. And basically test out whether that the latency that you see, the delay between on the ground operations and what we see in space would have a significant impact on being able to carry out those operations. And so, obviously this is kind of the first step of, eventually being able to develop the capabilities to do surgeries on, astronauts as they're in transit or in long duration space flight scenarios in a situation where you don't have a doctor on board or you don't have a doctor with the right medical expertise to be doing those operations. So again, this is the first step, but it's exciting to be taking this first step and seeing what works well and what doesn't, and what we might need to develop more in order to be able to use this capability in the future. And really the things that we're seeing for this type of application in space are very, very applicable to remote areas on Earth, where again, you may not have the right surgical expertise to be carrying out those operations. And so the potential to, to develop these technologies that has benefits everywhere is very exciting.
Thank you.
Thank you very much. If there are no other questions at this point, then we will jump into the researchers portion of the webinar. So with that, I would like to introduce Dr. Hema Ramkumar. She is the founder and CEO of Oculogenics, and she will discuss a gene therapy investigation that is launching in hopes to prevent or even reverse vision loss from the millions that are affected by age-related macular degeneration. I would also note, too, that after Dr. Ramkumar is done with her overview, that she has to run away because she is a working doctor. So if there are any questions from the media that you would like to ask, I would recommend doing it while she is still a part of the webinar. So with that, I pass the torch.
Thank you so much, Patrick, and thank you for the opportunity to be on the webinar today. So Oculogenics' mission is to improve vision for patients with retinal degeneration and age-related vision loss. And we have developed an investigational therapy that preserves retinal function and prevents retinal damage. We're very excited about our upcoming space mission. And the objectives of our mission are twofold. One, to assess if spaceflight to low Earth orbit can be a good model of age-related oxidative retinal diseases, such as intermediate macular degeneration. And two, to see if our investigational therapy can prevent this previously reported retinal dysfunction from the oxidative damage associated with spaceflight. So what we're doing is we have pre-treated mice with our therapy and then taken baseline retinal photos and retinal scans and assess their retinal function. Then we'll be sending them in the SpaceX 30 rocket in the upcoming launch. They'll have about 1 month trip into low Earth orbit. And when they come back, we'll be taking retinal photos and do retinal scans on them. And with this data, we're going to analyze to see if there are an increase in macular degeneration related biomarkers and oxidative stress biomarkers after spaceflight, and to see if our gene therapy can prevent that. So the exciting thing about our mission is that it's the first mission ever to have an ocular therapy in mice that are traveling to space. And the second is it's the first gene therapy treated animals that are going to space. So we're very, very excited about this breakthrough program and the potential to develop a new model for very, very common age-related diseases that currently do not have good models on the ground. And so we're excited to have been selected as the Technology and Space Prize winner during the Mass Challenge Accelerator a couple of years ago, and to have our research sponsored to have a big benefit for a very common disease on Earth, but then also hopefully with implications to improve our ability to travel to further areas in space.
Fantastic. Thank you so much for the background on your investigation. I see that we have a question from Will Robinson-Smith.
Yeah, thanks, Pat. And thanks for the overview, Dr. Ramkumar. Couple of questions to you. How similar are the optical systems of mice to humans, as in, how much of a one-to-one system, translation will there be? And then secondarily, will the astronauts who are on station have any interaction with the mice themselves, or will they kind of be quarantined in their own little pocket of the station, just kind of chilling there until they're brought back down when the Dragon departs? Thanks.
Thank you for your question, Will. The first question is about the similarity between mice and humans. While we've evolved a lot since becoming mice, our retinas are actually very similar. While we don't have a macula, we have all the same retinal cell layers as mice. And the standard investigational therapies for oxidative stress and oxidative damage are using mouse models. So very similar, I would say, with some differences. However, with the retinal imaging equipment that we have, we're able to detect changes in biology on retinal scans similar to what we see in humans on mice and with spaceflight. Hope that answers your first question. So I was saying many similarities, but just the only difference is that there's not a macula, and that's the key difference. And the second is the astronaut role in taking care of the mice. So the astronauts will be fully in charge of animal husbandry. And so all of the surgical treatments or things that we've done happened before, and all the therapeutics have been given before, but the astronauts will be involved in basic animal husbandry and care throughout the mission.
Thank you very much. Great question, Will. And actually, I had a quick question on my end too, where you mentioned that this was a project that derived from the Mass Challenge Startup Accelerator Program and the Technology and Space Prize in association with the ISS National Lab and Boeing. But the question I would have is, prior to that Technology and Space Prize, had you ever thought that spaceflight would have been an avenue of opportunity for you and for the can't even say it right now. I'm going to let you say
Oculogenics.
There you go. You can't take me anywhere.
So actually, that's a great question. You know, I have been researching mouse models of macular degeneration for over 15 years, and I had no idea about the ability to utilize space flight to mimic the biologic pathways that we see in patients with macular degeneration until I was in the Mass Challenge Accelerator. And that's because a lot of the amazing work that the NASA Space Bio program has done in the last few years in carefully describing the changes in the retina seen in mice that go to space. And that work by Mao's group showed that we are turning on macular degeneration related genes, activating the same biologic pathways, and then we have a 10% loss of retinal cells and 30% loss of retinal function. So all of this information I found out only because of the Technology and Space Prize opportunity and the fact that the Space Lab, National Lab, reached out to me during the Mass Challenge Accelerator. And I think that, and that's why one of the goals of our project is to just get more validity and data around the fact that spaceflight is a great option to create a model of a very difficult to model disease, which is, an age-related oxidative damage.
Fantastic. Well, again, thank you so much for taking the time out. I know that you are very, very busy with, again, with your day job. So I want to be respectful and mindful of that. And so if there are no additional questions at this point, we'll move on to our next speaker. So we have obviously the first one for this team, but then now we're going to be moving into Dr. Paula Grisanti, and she and the team over at the National Stem Cell Foundation are in the process of launching their 6th investigation to the orbiting laboratory, focused on 3D brain models derived from induced pluripotent stem cells of patients with Alzheimer's and Parkinson's diseases, as well as primary progressive multiple sclerosis. A lot of big words, and since I've not been able to do a great job of saying big words today so far, I'm going to hand it off to Dr. Grisanti, and she might be able to tell us a little bit more about this investigation.
Patrick, I'm unable to start my video.
Well, let's see. Worst case scenario, or we still have you on? There we go.
There we go. There we go. So since 2018, we have been sponsoring the first bicoastal collaboration between leading experts in the fields of Parkinson's disease and progressive MS in a first-in-kind study of neurodegeneration on the International Space Station using cells derived from using organoids developed from the cells of people with Parkinson's disease and progressive MS. On our next flight, not this one, not SpaceX 30, but on our next flight, we'll also be adding organoids developed from the cells of people with Parkinson's disease. I'm sorry, with Alzheimer's disease. In space, because you can see cells interacting in a way not possible on Earth, you get a much better understanding of how these disorders develop, and it opens the door to new cell and gene therapeutics that for diseases that affect tens of millions of people worldwide. We initially started this study thinking that in space, watching these cells interact, we could see a point at which things started to go south in the development of those disorders and intervene with a new cell or drug therapy that would prevent that from happening. What has happened is that we have developed the first human organoid models of these diseases in space, where they age more rapidly than they do on Earth. In space, you can observe in a period of weeks or months what might take you years to see on the ground. So in these diseases of an aging population, these cells are very valuable. These organoids can be instrumental in research and clinical trial assessment in all kinds of applications for us to begin the process of intervening with new therapeutics.
Fantastic. Okay, let's see. Trying to see if we have any. I have a quick question on my end. So again, as we intro this is your 6th investigation that's going to be launching. Can you maybe talk a little bit about how it is that your team has evolved your respective projects over the years, and what you've learned basically just about trying to package up an investigation and launch it to the space station?
We actually, we're funding 2 separate organizations. We were funding Jean Loring at Scripps Research, and we were funding Valentina Fossati at the New York Stem Cell Foundation Research Institute. Jean was working on a induced pluripotent stem cell intervention therapeutic for Parkinson's disease, and Valentina was working on cell crosstalk around oligodendrocytes, the cells that produce myelin, that are the protective coating around nerve fibers that allow your brain to talk to the rest of your body. And I thought all these years ago that they would make a great marriage. If you've got a researcher in California who's trying to do make cells do what you want them to do, and you've got a researcher in New York who's trying to keep the cells in the neighborhood happy, that would be a great opportunity for them to join up and potentially create this study that could be conducted on the International Space Station, where you have this opportunity to see things in a way not possible on Earth. And that is, in fact, what happened. It's been a wonderful marriage. So we're working with Valentina Fossati and Scott Nagel at the New York Stem Cell Foundation Research Institute, and Jean Loring, who is Professor Emeritus at Scripps Research. And this study, over this 5-year period, has developed into something that is a reality for these discoveries that are now driving innovation into potential new therapeutics.
And I had the privilege of spending some time with you and the team a few weeks back out in New York. And one of the things that was also communicated that's a little bit different about this mission is that I believe that you're able to get some different types of real-time data, because you're going to be using a microscope of some nature that previously you had not had access to. Is that something that you can maybe expand a little bit more about?
Yeah, we're looking forward to having access to the Keon's microscope on this flight. We're doing drug challenge on this flight, which is looking at drugs to develop and assess maturation on the International Space Station and perhaps promote it. The microscope will allow us to do a much more in-depth search. And then on our next flight, we'll be adding drug screening in addition to the Alzheimer's organoids that we're using. And again, fluorescent microscopy is going to make a huge difference for us.
Thank you so much for the background on that, and continued success to the team. And we look forward to future missions or future space flights on future missions with your team in the future. So a lot of future. With that, I would like to move on to Marc Elmouttie, who is the research group leader for CSIRO, an Australian government agency responsible for scientific research. And he will discuss the multi-resolution scanner that will use the Astrobee autonomous free-flying robotic system on the space station. Marc, good morning. And by the way, too, I think that you are actually in Australia. So I wanted to give a quick shout out to say thank you for being up so early to be with us today.
Oh, look, thank you so much, Patrick, to you and ISS National Lab for the opportunity. Yeah, it is 3:30 in the morning, but I'm trying to stay as coherent as possible. So please pull me up if I slur my words or anything like that. But look, thanks again. Yeah, my name is Marc Elmouttie. I'm based in Brisbane, Australia. I work for CSIRO. This project and the payload that we've developed is a collaboration with Boeing. And Boeing is actually one of the key strategic partners of CSIRO. And our Houston-based Boeing lead, Connie Miller, is on the call today as well. So hi, Connie. Just to give you some background on CSIRO, for those of you that are maybe not familiar with it, it's Australia's national science agency. and we're tasked with solving problems for industrial and societal benefit. We have expertise and resources across the array of sciences, and they include, the life sciences, space and astronomy, environmental sciences, et cetera, et cetera. But for this payload, we've actually tapped into our expertise from our robotics and our mining technology research groups. And the payload we've developed, as you said, Patrick, is called the Multi-Resolution Scanner, or we call it MRS for short. And it's a 3D scanning payload. We refer to multi-resolution because it's actually capturing data from a range of sensors with both low and high resolutions. And the data is then combined or fused, as we put it, to deliver a number of digital products. And those are mainly an accurate trajectory of the position of the orientation of the robot in which the payload is housed in, a lower resolution 3D map of the environment, and a higher resolution, narrower field of view 3D map of regions of interest as the robot is moving through space. The payload uses algorithms that CSIRO has developed to perform this fusion and this processing. One of the algorithms is known as Wildcat SLAM. So it's a world-leading simultaneous localization and mapping algorithm. And it actually generates the accurate trajectory information that I was talking about, as well as the map of the environment. So Wildcat SLAM actually achieved award-winning results at the DARPA Subterranean Challenge, which occurred a couple of years ago. And it's also been commercialized to support drone-based solution for the mining industry and other industries. And the other algorithm is a patented algorithm known as stereo depth fusion, or SDF for short. And once again, that was developed originally for the mining industry, and it generates high-resolution 3D maps of those regions of interest. So it's actually been applied now for mining and industrial engineering applications, but we've also been working on it in collaboration with Boeing for a number of years. Boeing have a very interesting project known as the Inkjet project based in Seattle, and they are developing an autonomous aircraft paint and livery deployment system, and SDF is being developed as part of that research. Now, around 5 years ago, Boeing and CSIRO began discussions and planning to develop a payload that would be based off these algorithms, but target some specific space applications. And in particular, the first one was interior vehicular scanning. And the main use case was for autonomous spacecraft operations. And that's actually what we're demonstrating on this mission. But there's also future use cases that we're targeting. And in particular, they are exterior vehicular scanning for defect detection or hull damage, and also the off-world scanning, such as for rover payloads. So we've tried to design the payload with those three use cases in mind, and for it to be scalable and extensible to support those three use cases. So the payload has obviously been designed in close collaboration with NASA, and in particular, NASA Ames's Astrobee team. And the reason for that is because the payload will actually mate or plug into one of the Astrobee free-flying robots on the International Space Station. Now, I'm sure Astrobee is featured on previous webinars, but just in case, I've sent through an image of our payload mated to an Astrobee. And you can see that on the screen now. So you can see that the Astrobee itself is a cube-shaped robot, roughly a foot or 30 centimeters on its side. And you can see on the top there a shoebox size, a black box with a couple of lenses taped over there. And that is our payload occupying the payload bay of the Astrobee. And some NASA staff have sort of referred to this as giving the Astrobee spectacles or extra eyes to map the environment. So the payload consists of a number of sensors, some of which you can see clearly in the image. They include 3 time-of-flight sensors, an inertial measurement unit, and the stereo camera. And obviously there's onboard compute to perform this processing. So the goal of the mission is to demonstrate that by using our payload, the Astrobee can successfully map the environment of the Kibo or the GEM module on the ISS. And the demonstration will actually prove that the payload can support robots like Astrobee or other robots to undertake spacecraft caretaker operations. But the other interesting thing is, aside from those science objectives, this one of the milestones we're trying to achieve is this is the first payload that's actually occupying both payload bays of the Astrobee. So typically the Astrobee supports payloads which are essentially half the size of what you see on the screen there. And so NASA advised us early on in the planning of the project for operational reasons that we should consider designing a payload that occupied both payload bays. And so if this successful demonstration will actually open up the possibility for future payload developers to use the Astrobees on the ISS in this configuration. So that's a summary of a project. I'm obviously here today representing quite a large team spread across multiple organizations. I'd just like to take a brief moment just to say thanks to my colleagues at CSIRO. And as I said, the project itself is drawing upon expertise from the Space Technologies research program at CSIRO, and that's Kimberly Clayfield, as well as the mining technologies and cyber physical systems research programs, and that's Huaguo and Navinda Kotager. But I've just sent a photo of some of the Brisbane-based team. And as I said, the collaboration is with Boeing, and they're spread across the US and Brisbane as well. But on that photo there, you see some of the Brisbane-based project team from CSIRO. We're holding our payload, which we affectionately refer to as Echo, and that was just taken prior to its shipping to NASA. And so one of the great privileges of being involved in this project has just been working with this wonderful team of engineers and scientists. So in the photo, you can see myself on the left, T. Molnar, going from left to right, Lauren Hansen, Matt Vanderwerk, and David Haddon, Roston Gable, Anna Campbell, and Paul Flick. And finally, I just can't speak highly enough of the NASA payload integration team, the journey that they've taken us on for the last 2 years, and Christian Garcia has led that, and it's just been a fantastic experience, as well as, of course, the NASA Astrobee, the NASA Ames Astrobee team that we're working very closely with, and you know, Jonathan Barlow and Jose Cortez and Jose Benavides and the whole team there have just been fantastic. So look, thanks again, Patrick, for the time, and thank you again to ISS National Lab for all the support. It's just greatly appreciated.
Awesome. Well, thank you so much, Marc. Quick question on my end too. And again, you talked about the many number of partners that are involved in this, whether it's NASA, Ames, Boeing, the Astrobee, your team. I mean, it truly is a worldwide collaboration. Is it maybe, can you maybe talk a little bit about, you know, how it is that space just seems to bring a lot of people together in the importance of being able to have different types of voices from, again, quite literally all over the world to make something like this come to fruition?
Yeah, thanks for the question, Patrick. And look, for quite a few of us on the team, particularly the Brisbane base, this is the first time that we've taken a payload all the way through to flight readiness and launch. So we can only now sort of fully appreciate how hard space is. And yeah, in terms of tackling those challenges, that multidisciplinary but also multi-organization approach has just been fantastic. I mean, the technical meetings that we have, the regular dial-ups we have, will have, you know, quite often 20, sometimes 30 people on the call, and they'll be spread out all over the world. So just being able to draw upon the expertise from those different parts or different organizations in different parts of those organizations has been fantastic. And it's something that CSIRO has done in all its science as well for all industrial applications, is really try to collaborate sort of internationally as well. We do have an active space research program at CSIRO, and one of the priority areas that we're targeting is in situ resource utilization, or ISRU. And that's another example where it's CSIRO working with NASA and the other space agencies to try to accelerate the TRL levels of some of the key technologies to meet that quite daunting challenge. And once again, you can only do that through that kind of collaboration that you mentioned, Patrick.
And I guess a quick follow-up to that too is, you know, you mentioned that this is kind of a validation, if you will, first of its kind. Assuming that everything goes according to plan, what will be next steps for your team on this particular project?
So yeah, as you said, the key target here is successful mapping of the Kibo module, and that may require one or possibly two missions to successfully complete that task. And we've already started discussions with Boeing and NASA for some of those other use cases. In particular, the exterior vehicular scanning or hull defect detection use case is something where the conversation has started. A bit more of a challenging problem in terms of the exterior environment and dealing with the radiation hardening aspects of that. And several of the sensors on that configuration obviously would be subject to those kinds of exposures. But that's probably the next step. And then I did mention ISRU, which is the rover-based application. And for that, we do have a test facility in Brisbane where we've been already starting to explore some ideas of what would this payload look like in that kind of rover-based deployment. I think I've just lost your audio there, Patrick. It could be at my end.
Oh, no, it's because I forgot to press the unmute button. So but again, I wanted to say thank you so much for taking the time to join us again, given the fact that you are, again, literally 3:30, 4:30 in the morning right now. So hopefully you'll be able to get some rest after this, and successful launch forthcoming.
Much appreciated, Patrick. Thank you.
Of course. All right, so we're going to move on now, and we're going to have a little bit of a BOGO. So first, we're going to have Chris Keno, who is the Managing Director for Space Exploration Operations at Airbus U.S. Space and Defense, highlight Argus, a new mechanical platform capable of hosting payloads outside of the space station. After Chris provides an overview, we will bring in Liam Kennedy of Sen Corporation, and he will give an overview on Space TV 1, a project that will be leveraging Argus. So Chris, let's start with you.
All right, Patrick. Well, thank you. I appreciate you putting this together, and thanks for everybody on the line. And let me say thanks to NASA and ISS National Lab as well for the ongoing support of us commercial service providers on ISS, and and how that all comes together in terms of continuing to spur this LEO economy that we're all working so feverishly towards. So I'm going to briefly address the payloads that we have flying up here on SpaceX 30. And I'm going to come at this from a little different angle, and that's the infrastructure perspective, right? Because I can't talk to the science, but I can talk to the hosting capabilities. So on this payload going up on this flight for SpaceX 30, we have our avionics box, which is basically the brains behind the Argus multi-payload carrier. And we have 3 different customer payloads that we're bringing up. One of which Liam will talk about once I'm done here in a minute or two. It's a high definition camera. Then we have some other payloads that are doing TRL raising for suites of instruments and edge computing architectures and these type of things. So we're very excited because this will be the first deployment of our Argus multi-payload carrier onto our Bartolomeo external payload platform, which is mounted on the ISS. So I think to explain Argus better, I probably need to give a little brief overview of Bartolomeo as well. So I have a couple pictures here, which I think will help move through this quicker. You can go on to the next one if you like. There we go. So this one right here, I just want to point out the location of where Bartolomeo is on the ISS and where the payloads will be hosted. You can see it's there on the leading edge of the station. Attached to the Columbus module. And of course, from this location, we have, what I believe to be very superior accommodations for payloads that Earth observation, deep space, anything with nadir viewing, zenith viewing, RAM, limb viewing. So that's to put just a little perspective around where the payloads will be hosted and the kind of environment they'll be in. If you want to jump to the next one, please. So this just drills down a little bit deeper, a little cartoon of what the platform there on the mounted to Columbus looks like. Those big boxes that show there, those are really graphical representations of the nominal volume we can host at each of those spots based on the airlock dimensions. And as you can see on the platform, there's 8 hard-mounted payload locations on Bartolomeo itself. 6 around the exterior edges on the starboard port and leading edge, and then 1 on the zenith side and 1 on the nadir side. And it also kind of shows a little picture there of what an Argus carrier might look like in terms of how this can be deployed to a single payload location on Bartolomeo and host up to 10 different payloads. So if you can go to the next one, I know we're running a little short on time, so I'm going to try to move quickly. To go a little further now to look at what Argus is in and of itself. So that it really consists of three different components. It has the base plate, which is where the payloads are mounted, which also has the Gold 2 adapter. That's the connector that we use. It's manufactured by Oceaneering that allows us to do robotic installation onto the platform and also makes the power and data connections back to Bartolomeo, which then power the avionics box, which is the third component. And from there, that powers each of the individual payloads, kind of modulates the power and data requirements to each of the payloads. Yeah. That's all I think I needed on that. So if you jump to the next one, I think was there one more slide? Was that it? Okay, that's good. We're good with that. So really what I wanted to say was, like, again, we're really excited that we're able to bring this capability forward. And in that infrastructure isn't near as exciting as a lot of the what we're hearing about so far, previous to this and the presentations after this. But I think it's really important to be able to provide that capability and infrastructure to support the ideas that come from industry and come from academia, and the type of things that are going to drive that development of that low Earth economy, and really drive us to the next levels as we move from ISS onto what's next. So with that, I just wanted to touch on the capabilities we have here that we're bringing up on flight. Now this comes up in several different pieces. The backbone launched on NG-20 about a month or two ago. And then with these payloads coming up on SpaceX 30, the astronauts will integrate the avionics box and the payloads onto the Argus backbone, which will then be translated out the airlock and then robotically installed on Bartolomeo for roughly 1 year exposure out in space, then brought back in and wash, rinse, repeat with another set of payloads. So that's a quick overview of the hardware that's going up. And Liam will talk more about the specific SEN payload after this. So if there are any questions.
All right, I'll zoom in, and if anyone wants to ask questions for you, Chris, obviously, hopefully they'll raise their hand. But hi all, yeah, I'm Liam Kennedy. I'm the Space TV Director for SEN. And I wanted to start off by telling you a little bit about SEN, because you may have never heard of us. And hands up anyone who, for them, it's the first time knowing that we are already streaming and providing live 4K views of the Earth and pre-recorded views of the Earth from our first satellite, which is called ETV-A1. And a little bit of history as for, well, how does that translate into what we're doing here now? Back in, I've been involved in sort of really being focused on live views of the Earth since really 2014. So we're going on 10 years. But little did I know that the company SEN, founded by Charles Black, the founder and CEO, were looking at bringing this capability to the world. And in fact, this is what we do. SEN's mission is to stream real-time videos from space to billions of people, gathering news and information about Earth and space and making it universally accessible to all. And our vision is to democratize space using video to inform, educate, inspire, and benefit all of humanity. Really think of the overview effect and the impact that can have on the world. A little bit of info about the Space TV-1 mission. So yes, as you heard from Chris, the Bartolomeo platform and particularly the Argus sub-platform, that provides location. Yeah, I've had my eye on this corner of the space station for many years. And when I first heard about the Bartolomeo capability, I think it may have been at the 2018 ISS R&D or maybe 2019. From that point on, we started having a conversation about how we could bring our capability to the place in space where At that time, human beings were, it was really the only place in space where human beings were. And here is where our mission, which is operated by our US organization called SEND-TV Inc., licensed by NOAA for remote sensing. We're bringing a 4K camera system designed to live stream 24/7. And that will have 3 cameras, Nadir, Horizon, and docking port. And I think if you bring up the first slide, you had a preview of this a second ago. That is a slide showing the Argus platform, the Argus plate, and our payload attached to that. And you'll sort of count up there, you'll see that there are sort of 4 cameras that are visible. There's actually another 2 further ones that are not visible on here. And we actually have backups of every single camera that we're sending up. So let me just switch on to the next slide. And I wanted to just go through those views. So these are just really simulations of the views. So we'll have a horizon view, and we expect that to be able to show some awesome 4K views of sunrises. Our cameras are sensitive enough to work at night, so we expect to be able to capture auroras in real time. The Nadir view camera is shown there. That's actually Houston, for any that are very clever about recognizing cities from space. That's giving an example of the field of view as we're going over Houston. And then finally, a very exciting camera view that we have is pointed at the forward docking port, PMA2. So we'll be able to capture all of the extraordinary dockings in 4K going to that forward docking port. So, a little bit more info about the cameras. In fact, you can switch back to, you can take away from that one, come back to me, and there we go. And yeah, so very quickly, all of those cameras, we are recording up to 20 hours of each camera on the payload itself. And we're very happy to be able to use the Airbus the data communications connections through everything that's coming from the space station, be able to get access to our content. As I say, we'll be able to stream one camera throughout the day, and but we'll be able to switch between any of the cameras at any time that we need to do that, and also downlink pre-recorded content as well. And that means for the very first time, we will have uninterrupted access to events that are going on the space station through these cameras. We will be somewhat immune to loss of signal events because we're capturing in the payload itself. And I'd just like to finish up by saying thank you so much to Airbus, to Chris Connell, and all of the staff at the Houston side of things who've worked with our team, our engineering team, and our general manager, that's Christy Stockey, and just helped us get through all of this work, and especially the ISS National Lab. I had no idea that this kind of thing was going to be possible, and it's only possible because of NASA's real support for commercialization of the International Space Station. And we look forward to bringing this to the world very soon. If you go to send.com, you'll be able to follow along from there. But thank you.
Awesome. And I'll say, Liam, thank you. And that's what we do.
Indeed. Bringing people together. Quick question on my end. So, this is also, a great representation of how it is you can use an external facility for camera or sensing capabilities. But Chris, maybe this is more for you, is, why else would a researcher want to leverage the outside of the space station as far as like the environment itself?
Well, so that's a fantastic question. And, I think the answers that I could come up with are probably the more obvious ones. And what's really exciting about that question is the answers that we haven't thought of yet, right? That industry can bring forward into this, because, having these opportunities especially now as more and more, there's more opportunities to get to orbit, more opportunities to do these type of payloads. Who knows what industry will drive? But you're right off the bat, I would say your obvious Earth observation, looking into deep space. I believe it's a great test bed to start to work on, advanced manufacturing techniques in space. Maybe to me, it's an opportunity to prove out, viability and scalability of different types of manufacturing approaches before, commit to next phases. So the answer really is it's whatever the payload community come up with. If it fits within the volume and the power and the data restrictions we have, it can fly. And that's one of my favorite things, is talking to potential payloads and see what it is they're trying to do. So that's kind of an answer.
It's kind of an answer, and I will take it. But I mean, to your point, if you can withstand, being on the outside of the space station in that extreme environment, then you can pretty much withstand just about anything. So that's exciting stuff. I see that we have a question from Will Robinson-Smith.
Yeah, thanks, Pat. Not sure if this is better for Chris or for Liam, or maybe the both of you, if Chris is still there. But in thinking of additional applications for this technology, you mentioned, looking at other opportunities. Have you had conversations, or might you have conversations down the road with some of the CLIPS providers as far as being able to bring these camera systems onto some of the lunar landers bound for the moon? I mean, I know we were pretty captivated by the images sent back by Astrobotic and Intuitive Machines on their journeys. So just curious if that's in your future, or you might have those conversations down the road. Thanks.
I'd be happy to share a perspective that I've got, Chris, but
no, I defer to you on this.
Yeah. So really grateful for the question, Will, because it allows me to expand a little bit about on where we're going. We have our current satellite, ETV A1. We're moving to the space station soon, Space TV1. But we are planning on having our camera system on all places in space, specifically where there are going to be human beings. And also, we do have a dastardly plan to get to the moon too. So yes, I think if ever you're involved in any of those missions that are doing things like that, we want to bring people of the Earth in ultra high definition to all of those missions. And that also means that we have to worry about, well, how on earth do we get that data back to the Earth? So hopefully we're going to be one of those organizations that is pulling for the capabilities of doing that, both at an infrastructure level in terms of the data bandwidth, but also just in thinking about how can we do that. So this is just one example of what we're going to be doing. We'll be up on the space station, hopefully for 3+ years. And during that time, we may even, with Chris's support, launch new payloads that have additional camera capabilities along the way. So expect more in that area. And hopefully that helped answer that question.
I would only add that, that model applies to almost any technology that you need to develop to head, from LEO to cislunar to lunar. So again, it comes back to the payload customers, the science, what has to be proved out. I think this is one of the capabilities that's required. That's it.
Awesome. Well, thank you very much. And Liam, I would love to go out there and not look at dastardly things, but more of awesome things in the future with you and in the team. So again, thank you so much. Best of luck with this launch, Chris, for your team to help to validate this capability and leading to additional collaborations in the future.
Thanks, Patrick.
Thank you.
All righty. So next up, please welcome Michelle Lucas. She is the founder and CEO of Higher Orbits, a nonprofit that aims to inspire students into STEM-related careers and is also supporting 3 student-led investigations on this mission. Michelle, happy Friday.
Happy Friday. Thank you so much for having me here. I am just over the moon to be able to share with you about some of these student experiments. Patrick, thank you for inviting me to be part. So this is one of those, now for something completely different. Our PIs is not actually me. I am not the smart one. The smart one is our students, and I should say smart ones are our students. On this flight, we have 3 student-led experiments. This will be our 18th, 19th, and 20th experiments fly to the International Space Station through a program that we call Go for Launch. Our Go for Launch program is a multi-day space-inspired STEM event that we run all across the United States, where students in grades 7 through 12 get to work with an astronaut for the entire event. And through the course of this, they design an experiment that could fly to the International Space Station. Now, as opposed to taking years to develop this, which, let's face it, is how we're used to working with science, they get a couple of days. And at the end of the event, they will be able to present their idea. A panel of judges selects them as a finalist, and we have an overall series winner who has their experiment flown to the space station. This flight is carrying 3 series winners from across the country. Could I have my slide, please? We have 3 teams. Our team from Midland, Texas is on the top left. That is Team TARS. We have Alyssa, Ulysses, and Catalina. On the top right, we have Team Space Shell 6, Sierra, Zara, Taylor, Siri, Claire, and Evelyn. And at the bottom, we have Team Phoenix. Actually, I think we actually goofed up our photos here a little bit. I'm sorry. Our team from Phoenix 229312, I sent over the wrong photo, so my apologize. But we have Zara, Devin, Rookie, and Anthony. You can drop the slide. These 3 experiments will be housed in the same facility thanks to the work with Space Tango. Now, the students conceive and define these ideas, but they don't actually build them. What they do is they're going to take their ideas and they work with Space Tango, who physically builds these experiments for us. Our Midland experiment is about fruit flies and bone density. So you're thinking right now, okay, fruit flies don't have bone density. Correct. However, there are some elements that we can evaluate within fruit flies by changing their diets, determine if an iron-laden diet helps with bone density and microgravity. So the fruit flies will fly with, half of them will have a supplemented diet that has iron in it. The other half will not. We will evaluate those 2 against each other, as well as against some fruit flies here on the ground. Our second experiment is from Team TARS. That was from Team TARS. Our second experiment is from Team 229312. And by the way, the students all come up with their team names through a variety of ways. Their experiment is the effect of radiotrophic fungi on satellite components. They're expanding on knowledge that we have gained before about how fungus grows more rapidly in space, and there is some fungus that helps block radiation. And so they are going to evaluate if this fungus can better block radiation on some electronics as a way of, instead of having to harden all of our electronics, perhaps we could use some fungus. The third experiment is from Team Space Shell 6 from Sault Ste. Marie, Michigan, and they are evaluating cells in space. They're going to take a look at the cellular CO2 production in microgravity and see how that changes. All of these experiments are truly student-driven. We have Space Tango to help them with the engineering elements and to help them further their designs. But at the end of the day, these are all student scientists. We're thrilled to be able to provide this program to students because it helps them build teamwork and also gives them the opportunity for project-based learning. And we get to add to the repository of science that comes from the ISS through the ISS National Lab. Students are super excited for launch whenever it happens this month. We all know how rocket launches are whenever it happens. And we hope to be able to have most of them at launch. So go Falcon 9, go Dragon, go science and space.
Awesome. Well, thank you, Michelle. A couple of quick questions on my end. The first one being, you mentioned obviously that Space Tango is the hardware partner, but then you also have an astronaut that kind of helps to usher them through the fun of trying to configure an investigation to the space station. But do the students themselves, do they specialize in any one thing, or do they have a chance to dabble in all aspects of kind of, developing a project?
Patrick, I love that question. And yes, thank you. They do have an astronaut to serve as a mentor for the entire event, which alone, that is pretty amazing to me. But they get to pick in general. Now, I have to give them some constraints. I tell them that injuring the astronauts obviously is always bad. We don't do any animals with backbones. They're not allowed to explode anything. But then they are given free rein to come up with whatever will fit inside a 1 to 2U cube lab inside ISS. So that's why you see such a wide variety of experiments. And it's why we've had such a wide variety of experiments across the years as we've flown experiments since 2017 from students.
Awesome. I see that Jim Siegel has his hand raised.
Okay, can you hear me?
We got you, Jim.
All right, thank you, Patrick. And it's a pleasure to be here with you today. And thank you for doing this particular webinar. I have a question about the fruit fly experiment.
Yes.
So are these fruit flies kind of, do they have to be loaded at a certain point, a minimum point onto the spacecraft? And how many fruit flies are going up in each group? And are these fruit flies going to come back to Earth, or what happens to them?
So that's those are all great questions. We do have a little bit of a late load, not truly late load. We do have cold stowage going up. Space Tango has worked all those elements. The specific fruit fly, and Patrick, you had challenges with words earlier. Now it's my turn. I tried to stay away from the big words. Drosophila melanogaster. So there will be 2 different colonies of fruit flies. I'll be honest, I am not positive exactly how many are in each. But with them being in space for 4 weeks, they will also have offspring. We've made sure that the habitat is large enough to contain all of those. The experiments are going to come back to Earth, and so we will get some visual data while it's in space, but we're going to be able to get the most data when we bring them back here to Earth, and we are able to pull samples from the fruit flies to test their iron levels.
Okay, thank you very much.
Thank you for your question.
Well, Michelle, I am happy to hear that my being tongue-tied earlier is something that can be passed along to other participants as well. But again, it's always great to work with you and the team. Great to see a picture of an old friend in Greg Johnson too. So I'm assuming that he was the astronaut that was assisting some of the students this time around.
Yeah, so we have different astronauts at all of the events, but Greg Box Johnson was the astronaut at the Midland event. And so former ISS National Lab director, I guess it was Cases at that time. So we're always thrilled when he can come and share his expertise of not just space flight, but science and space with the students.
And then final question for me is the obligatory, if there are people that are watching that are interested in a little bit more about how they or their communities can get involved in this, how can they get involved?
Well, Patrick, I appreciate that. You can take a look at our website, Higher Orbits with an S, like the proper term, .org, and you can see what communities we will be in this year. We do have some of our Go for Launch events already scheduled all across the country, but we also are always looking for new collaborators, whether that is a host city who would like to bring us there, scientists who would like to work with our students, or companies who would like to make sure that students have the opportunity to do this project-based learning. If you click on Contact Us on the website, the only other living creature in the office has 4 legs fur, barks at FedEx, you'll get me. So feel free to drop us a note. We love to collaborate with anybody.
Awesome. Well, thank you so much, Michelle, and we will see you in Florida in the not too distant future.
Thanks, Patrick. Go SpaceX 30.
Go. Okay, so up next, last but certainly not least, will be Jordan McCaig. Jordan is a graduate student at Georgia Tech, and she will discuss a NASA-sponsored investigation, genomic enumeration of antibiotic resistance in space, or as NASA and the space community like to do, since we live in the world of acronyms, we like to call this GEARS, which is why I can't say it. So Jordan, let's learn a little bit more about microbes in space.
Yeah, thank you for the introduction, Patrick. So yes, as mentioned, I am part of a group that is studying antibiotic resistance in space. So we can go ahead and jump right in if we want to get that first slide up. So wherever humans go as we explore through space, our microbes go with us. They are inside our bodies, they are on our skin, and they are all over the surfaces that we inhabit and the air that we breathe. Most of these microbes are harmless, and some of them are actually beneficial. However, some of them can also make us sick. On board the International Space Station, there is routine microbial monitoring that is conducted to understand how these microbes change and may influence human spaceflight. This accomplishes a few goals. First, it helps us understand what microbes are there and what health risks they may pose to astronauts. It also helps us understand how life forms from Earth can adapt to the unique stressors of spaceflight. This routine monitoring has actually indicated the presence of microbes that are part of the Enterococcus genus, which is seen on the left in that colorized green image. Enterococci are a common member of the human gut microbiome and are often harmless. However, they can sometimes exhibit high levels of antibiotic resistance, which can make people sick. In fact, antibiotic-resistant enterococci are a leading cause of hospital-acquired infection. To make matters worse, bacteria often exhibit an increased level of antibiotic resistance in space. This poses a risk to astronaut health, especially when you consider that astronaut immune systems often become compromised by spaceflight conditions. So the GEAR study is part of 3 different investigations to better understand enterococci in space. Specifically with GEARS, we are interested in learning more about how often enterococci are transferred to space and how they're spread through the spaceflight environment. So we can go ahead to the next slide, which will show the workflow for this study. This monitoring will be accomplished by integrating an antibiotic-resistant screening step into that routine microbial surveillance that I mentioned. So this thing on the left is called a contact slide. It's basically a rectangular-shaped petri dish. That yellowish material inside of it is called agar, which is a solid substance that microbes can latch onto and eat. Our team prepared special contact slides that contain an antibiotic that we know that enterococci can become resistant to. So the astronauts will swap surfaces and plate it on these slides. And you'll get these colonies that are growing in kind of different colors and shapes and sizes. Since there's an antibiotic within that agar, anything that grows on this plate is resistant to that antibiotic at that specific concentration. So we will extract and sequence the DNA from those colonies. The astronauts will do this all on orbit, and this will be enabled through the presence of a nanopore DNA sequencer on the station. So in this third image, you can see one of the astronauts proudly displaying the sequencer. The actual sequencer itself is that little gray rectangle that is underneath the computer monitor. And this is a really cool technology innovation that enables direct sequencing of DNA within a few hours of collecting your samples. Being able to do this direct DNA sequencing on orbit will demonstrate our ability to conduct microbial monitoring of these pathogens that could be potentially risky on station. And as human spaceflight missions continue to get further away from Earth and go on for even longer, this will be extremely useful when we can't necessarily send things back to Earth for quick analysis. For the purposes of the study, to maximize the science yield, we will also be sending these bacteria back to Earth. There, they'll be more extensively studied to better reveal their genomic features and understand the risks that they may pose to astronauts. So this investigation is the first attempt to directly focus on the specific human pathogen in space and try to figure out the frequency and the identity of antibiotic-resistant organisms on the International Space Station. As we continue to proceed through this exciting time of crewed spaceflight, this sort of knowledge will be really useful for trying to figure out what sort of things may make astronauts sick, optimizing astronaut health, understanding what happens to microbes in space, and potentially making plans for potential countermeasures. So with that, thank you, and I'm happy to take any questions.
Jordan, that was a fantastic overview. Quick question on my end to kickstart things. So was there any prior microbial investigations that your team was looking at that helped to influence this particular investigation?
Yeah, absolutely. So some previous studies were mostly focused on doing general microbial surveillance without specifically looking for that antibiotic resistant stuff. And this indicated just by doing the analysis of those results that we thought that there were bacteria there that could have that antibiotic resistance. So the thing about this study that's making it innovative is having that direct antibiotic screening step so that we know that anything that's growing is resistant to antibiotics.
And I think that you mentioned too, you mentioned 3 steps. Now, does that mean that there's 3 steps associated with this investigation, or that this is the first of potentially multiple flights that your team will be working towards?
Yeah, so this investigation will actually span multiple flights with just multiple bringing plates up to the International Space Station. And then there are also 2 other investigations that our team is working on. The second one is called Interrogate, which is a way to directly study the evolution of enterococci on orbit. And then the third one will be called Aries, which will be something where we compare the isolates that we get from the GEARS study to other isolates from this species that have been previously found in other places on the space station and on Earth.
Fantastic. And in the last question of the Denise that I would have is, you are a graduate student right now at Georgia Tech. What's it like to know that as a grad student, you have the chance to be part of a team that is launching investigations to the space station? I have to imagine that's pretty fun in and of itself.
Yeah, it is awesome. I really enjoy it. I'm really looking forward to hopefully traveling to the launch soon and getting to actually see the science that I've been working on for a couple of years go to space. It's really a dream come true.
Awesome. Well, Florida in March is not necessarily a bad place to be. So we look forward to seeing you then. With that, I will pause to see if there's going to be any questions from the media. And what I would also throw out too is if there are questions, whether that be of Jordan or of the representatives that have been on that have joined the webinar today. If there's any questions from media, then we're happy to go out there and take those right now. If not, then we'll get ready to shut things down. Okay. Well, oops, sorry. Hold on. I see that Jonathan again has his hand up. Sorry about that. Jonathan, fire away.
No worries at all. My question is for either Davide or Heidi. It seems one of the advantages of research on the ISS is that biological processes move more rapidly. You can view on the ISS in months what it might take years on Earth. My question is, what speeds up those biological processes? Is it microgravity alone? Is it radiation? Is it all of the above? Or is it other factors unknown? Thank you.
I can answer to this. Thank you for the question. That's really interesting indeed. What I believe is most of the effects are due to microgravity, with less extent on board of the ISS due to radiation, because the radiation is more a cumulative effect that we do not really observe to great extent during our 30 days payload missions, for example. So microgravity has more impact on our biological experiments. And microgravity is an accelerator of conditions indeed, and makes everything happen faster.
Thank you.
Davide, or I'm sorry, Heidi, is there anything that you'd like to add to Davide's response?
No, I think that was fairly comprehensive.
Okay. Well, speaking of comprehensive, I think that this was a very informative webinar, and I hope that those that were able to join us were able to get quite a bit from it. So with that, I would like to thank all of the panelists for taking time out of their busy days, including those, again, like Marc, who are literally on the other side of the world. I would also like to thank the public and the media who took time to join today's webinar focused on R&D, launching on board NASA's SpaceX CRS-30 mission to the orbiting outpost. As mentioned, we are looking at launch no earlier than mid-March from Cape Canaveral Space Force Station in Florida. For up-to-the-minute information, please follow at NASA or at NASA Kennedy via social platforms, or follow the coverage on NASA's International Space Station blog. Again, we hope you found today's webinar to be informative. And with that, it has been a pleasure to be with you. On behalf of the ISS National Lab, again, I am Patrick O'Neill. Go Dragon and go Science. Goodbye.